And our next speaker is Lina de Nijs from the Pelagic Freezer Trawler Association. And she will be speaking about improving fish welfare in pelagic trawling, observations, challenges and opportunities. Welcome.
Is it working now? Yes. This sounds much better. So, yeah, well, enough about flatfish. We're talking around roundfish now. My name is Lina, and I'm working as a fisheries researcher for the Pelagic Freezer Trawler Association based in the Netherlands. And Michelle asked me to tell you something about the work that we've been doing the last couple of years. So, my first introduction was when I started working on fish welfare, I could clearly remember my first ever wild-caught fish, which was a mackerel on the west coast of Sweden during a summer holiday. I was very happy. I caught a fish, as you can see, but the fish does not seem to be very happy. So it moved, and I got really scared. My parents laughed a lot. Well, we unhooked the fish, we put it back into the sea, but at this time I was only 10 years old. I was only thinking of my own pleasure and not how the fish experienced it. But that changed a lot since I've been working on fish welfare. Well, our members are specialized in pelagic freezer trawlers, and they fish for commercial consumption, human consumption mainly. And our target species are herring, mackerel, horse mackerel, blue whiting, and greater Argentine. Well, it's already tipped upon today that in the beginning of welfare research, there was not really a focus on wild-caught fisheries, but that has been increasing the last couple of years. So that also triggered us to start working on this subject. Well, from catching to processing, a brief way through the system. We first start with capturing the fish. The fish is aggregated in a cod end, so there is a risk of hypoxia. And there are several places during the catching and processing process where poor welfare could occur, or the results of poor welfare could show. So the first step was during the capture, where the fish is aggregated in the cod end, and possible hypoxia could occur. So we did oxygen consumption measurements inside the cod end. The next step is during hauling and bringing the fish on board. We use a fish pump to transfer the fish from the net onto the vessel. So there's a pressure difference and some sort of roller coaster through the pump to arrive on deck. So we've been assessing fish condition when the fish came on board. The next step is temporarily storing the fish inside refrigerated seawater tanks, and I will call them RSW tanks further on in my presentation. Their fish are temporarily stored before being processed in the factory. So we did some oxygen consumption measurements as well in the RSW tanks to see what happens there. Then the last step, processing of fish, happens inside the factory. And there's not a welfare risk there anymore, but it's a place where we check the quality of the fish. And the results of poor welfare could show there. So as a proxy for the quality, we did some pH measurements to see if we could check whether there was a quality issue. Furthermore, during the trips, we've been collecting all kinds of operational values to be used as later on as explanatory values. So the journey we've been working on is that in the beginning, when we were thinking about catch welfare, we knew very little about the situation on board. So we first started with an exploratory trip in 2018, just to see what would be feasible on board a pelagic trawler. Well, this was quite okay. We saw some opportunities, so then we started kind of in the middle of the catching process with fish condition scoring. We've been doing this on almost all commercial valuable species to have like kind of a ground truth or what we've been working on, because you need to have a starting point. And for us, the starting point was, in what condition do fish come on board? And how does this differ between species and maybe also fishing seasons? By that time, we concluded that some fish came on board alive, and by visual checks in the factory, we noticed that all the fish being sorted and processed was dead. So they died inside the RSW tank. This made us wonder what's happening inside the RSW tank. So therefore, we extended the protocol with oxygen consumption measurements inside the tanks, RSW tanks. And when we did that, we still had questions like, okay, we now know in what condition they come on board. We know what happens inside the RSW tanks, but what's happening prior to getting on board, and how does this show in quality? So therefore, we again extended the protocol last year, and we did also the O2 measurements, the oxygen measurements inside the cod end, and we did the pH measurements in the factory. So it's now commonly known that out at sea, stress isn't a single event for a fish being caught. It's accumulated during the capture process. And this makes it also very difficult to determine what impacts the welfare of the fish most. And it also makes us think, like, where is the greatest welfare benefit to be achieved? So that makes it very difficult, because you can think of one fish, and you can maybe make the world a bit better for that one fish. But if that means that you have to discard like maybe half of your catch because the quality is just deteriorated so much, then you lose commercial value. Therefore, you have to catch more fish. So that won't be sustainable solution. So a solution for one fish may be good for one fish, but not on population level. So that's a bit difficult, and therefore we seek guidance on how to measure fish welfare, because especially on board, conditions can be quite harsh. So for this research, we focused on condition and environmental condition scoring and environmental conditions experienced by the fish as indicators for fish welfare. All experiments have been checked by the ethical committee to make sure they were okay to proceed with. Well, then we walked through through the process with the system. First of all, we did oxygen measurements inside the cod end. An average tow of a pelagic trawler is about 100 tons at once. So we've been putting up 2 standalone battery-powered optical sensors that measure dissolved oxygen inside the cod end. You see those in the red circles over there and over there. They were placed at about 8.5 meters from the back of the cod end, a little bit further to the boat, 8.5 meters, and just behind the first catch sensor, which you can see here. We chose those locations because the one on the side, this one, there could be a possibility of oxygen-rich water being exchanged on the side of the netting. This one, we really wanted to be fully covered by fish, even with smaller amounts. So therefore, we placed it after the first catch sensor. Well, we wanted to check whether the system worked the way we hoped it would work. So we put in a camera to check whether this was actually the situation. You see one sensor over here. The other one was clearly, shortly visible in the middle. The footage was taken while the vessel was making a turn, so that gave us a short view on the sensors, because quite fast we were catching fish and the footage just turned black because there was too much fish. But this was exactly how we wanted it to be, so we were very happy. Then we compared it with the operational values we've been collecting, especially whole catch size. For the graph, you see on the left axis, the y-axis, there's dissolved oxygen saturation, and that's there you have to read the blue line, and the other ones are read from the right y-axis, and that's orange and green, temperature and pressure. Pressure being a proxy for the depth. And what clearly became visible was that it really was different for the bigger the hole you caught, the less oxygen was available in the netting. So with small tows, with only catching about less than 50 tons of fish, you see that oxygen does drop, but it doesn't come lower than about 60% or something. Stays above 50 at least. So that's not that impressive. You can see here it's a crossing point and there's a crossing point. That means here is when we start fishing, we get to depth, temperature drops and pressure increases, and there the other crossing is when we start holding. For average tows around about 100 tons, you see a more extensive drop in oxygen, reaching to about 35%. But especially for large tows, over 150 tons of catch, you see that oxygen drops to almost 0% saturation. So that was quite impressive to see that happening. Then the second phase was to do condition scoring on board when the fish came on board. So we sampled the fish when they got out of the fish water separator. There the fish goes into the gutter and into the tanks, and the water is pumped overboard again. Well, we've been using the ramp vitality assessment method. Please note that if the reflex was present, the fish scored a 0, and if it was absent, it scored a 1. The total score is then calculated by the sum of scores divided by 9, where the score of 0 means very lively with no impaired reflexes, and 1 being very impaired. So we've been checking how does this relate to the operational values we've been collecting. But first of all, the results from the ramp scores. We've been doing it for the different commercial fisheries of imported commercial species. And you can see that between the species, but also within species during different fishing seasons, there was quite a different, there were quite different scores. Where you can show that horse mackerel is quite a tough fish, and scores relatively low, while for example mackerel, troll mackerel, the scores are quite high, and especially for blue whiting, which is caught at a depth of over 500 meters. Only a few fish survive the ascent to the surface. So we've been relating it to the operational values, hold duration, the time the cod end floats behind the vessel, and the sample being taken after the end of fishing. And you can see that for hold duration, there's a slight negative effect on the ramp scores. You see ramp score on the y-axis. Meaning that the longer the hold takes, the less, the higher the ramp score, so the more impaired fish come on board. The second graph, which was the time cod end behind the vessel, that was the time that they stop holding, the net just floats behind the vessel, and the pump is being connected. We didn't really saw a real effect, but that was only a time range from 6 to 11 minutes, so the range is also very narrow. Makes it difficult to actually see a relation. But then not all fish can be pumped on board at the same time. If you have 100 tons, it takes a while to get it all on board. So we also recorded the sample time and how much time there was between taking the sample and the end of fishing. And there you see a very strong relation that the longer it took for the fish to get on board, the higher the ramp score in general, meaning that the fish has been spending more time inside a crowded cod end. Then the fish, we noticed that fish were still alive during condition scoring, but in the factory we saw that it was dead. So we knew that something was happening inside the RSW tanks. So we used a multimeter equipped with a dissolved oxygen probe, and we put it in through the depressurizing tube from the fish tank. So this is a depressurizing tube, which is used for the cooling process. Therefore, the system, we could run this experiment, but the normal operations were not interrupted. So that was very important. When fish entered the RSW tank, and the tank is full, they close the lid, and they start the cooling process, because it's very important to cool the fish as fast as possible to preserve quality. Well, I selected 2 holes where there was a huge difference, again, with the catch size. So if you look at, we had a catch of only 50 tons, and we've been scoring the fish which entered that tank to see. And by scoring those samples, all the fish were alive when they entered the RSW tank. So there was a high percentage of living fish. There you immediately see that within 10 minutes time, you see an extensive drop of oxygen to only, well, around 10% saturation. So there was a lot of oxygen consumption inside the RSW tank. If you look at another hole, which was a bit too big, we scored all the fish again, or we scored the samples entering the tank, and we concluded that all the fish we had scored seemed to be dead when they entered the tank. And there you clearly see that within the same time frame, which is 10 minutes, not much happened, but even within 15 minutes you see a slight drop in oxygen, but not so extensive as you see it here in hole number 5. For hole number 5, we can zoom out a bit more because we had an extended range of data collection, and there you even see that within 100 minutes you reach 0% saturation inside the RSW tank. Sadly, for hole number 7, the equipment had crashed, so we could not visualize it any further. This means that as long as there are still fish alive inside the RSW tank, they continue to consume oxygen until 0% saturation levels are reached. Meaning that within the tank, fish die from a lack of oxygen. Then we were in the factory checking for the quality, and we measured pH. We used the same fish for pH measurements as the ones we've been used for condition scoring. So by scoring the conditions, they also got a mark, which was a tie wrap, very colorful. And based on the color of the tie wrap, it was sample 1, 2, 3, or 4. And if they were alive during fish condition assessment, then they got a second tie wrap. Well, we've been measuring at 3 different locations inside the fish. And you clearly see that with pH measurements, there was not a big difference between fish being alive or being dead inside the tank. So that we can conclude there that it is not much happening inside the tank. They are not building up extra lactate inside the tank, but they're also not demolishing any lactate. And the lactate buildup is one of the responses for stress experienced, and it already starts at the cod end when they're being captured. We also tried to relate this one to the operational values, and there you see that hole size doesn't have a clear influence, but the whole duration seems to have slight positive effect, and that was what we didn't expect, but maybe this has something to do with if the hole takes longer, some fish may be able to swim along with the gear and also already start to break down the highest lactate levels, but that's just a hypothesis. So to conclude, in the cold end, there is could be severe hypoxia or even full anoxia. So that already influenced the condition scores quite a lot, but the condition scores also vary a lot between species. Then there is quite a lot of oxygen depletion. If fish enter the tank alike, there's oxygen depletion inside the RSW tank. And what we can conclude from the pH measurements is that lactate levels and lactate production already peaked at the lowest level of capture stress. So that's inspired us a bit to look forward, and that we can try to avoid long hypoxia and near zero oxygen levels while fish are still conscious. And we may be trying to work on a fast euthanasia cold end to make that faster and less stressful. And we've been collaborating a lot with Wageningen Marine Research, so working a lot with Pika as well. So that's what we've been looking into, going to look into for the future. So sorry, I'm a bit over time, but. Are there any questions?
We have time for one question. Hans?
Lina, thank you very much for this interesting piece of work. There's one brief comment I would like to make about behavioral observations. You have to be very careful with drawing firm conclusions about that. I fully agree that you do that, so don't misunderstand me. But what we know from experience that when we left cod and haddock in a bin without water, we could measure EEGs for more than 2 hours.
Yeah.
And the animals didn't show any response in behavior anymore. So what I recommend when you apply the ramp test, apply a painful stimulus to see if you can make the animal respond. Give it a stronger stimulus. That may help you in your...
Yeah, that's a good test for a good addition to.
Yeah, because this is the only
future
technique
research.
you can use.
Exactly. We've been looking into EEGs, but especially on board, it's very difficult. So the RAM score is the most practical one in the future.
But one element to it and give a painful stimulus that can help you.
Yeah, that's a good one. Thanks.
Thank you, Lina.